In this video, I'm going to describe some genetic nomenclature that's essential for appreciating and understanding various aspects of genetics. One of those concepts is the concept of ploidy. And this is relatively a measure of how many copies of a chromosome are present in a cell. So for example, if we take a look here at this mouse karyogram, which is literally a photograph of all of the chromosomes found in a metaphase nucleus of a mouse cell, we can see that there are two different objects that are labeled 1, chromosome 1. And each of those chromosomes, there are 2 of them. Those are actually, though that's not critical to know now, those are actually 2 pairs of sister chromatids. You can ignore that for now if you like. I'm just being explicit. And so ploidy is defined at metaphase when scientists and especially cytogeneticists can take photographs of chromosomes when they're compacted at metaphase. If there are 2 homologs, there are 2 in this case. There are 2 copies of each chromosome, 2 of chromosome 1, 2 of chromosome 2, 2 of chromosome 3. That is diploidy, or 2. At metaphase, there are 2 copies of every chromosome. That's also true outside of metaphase. This is just the phase in the cell cycle when we can observe chromosomes. So this is the stage at which ploidy is determined. It's important also to understand where those 2 chromosomes came from. Fertilization. In a diploid organism, the sperm carries 1 copy of every chromosome, and the egg carries 1 copy of every chromosome. And when the 2 meet and fuse to form the diploid embryo, then there are 2 copies of that chromosome. So the gametes are haploid. They have half as many homologs as the diploid embryo. What this means is that every chromosome has a gene on it. So for example, we could indicate the position of a gene just by pointing an arrow at chromosome 1. And because those are homologs, the 2 copies of those chromosomes, even though they're genetically different because they came from different parents, every embryo, every diploid cell has 2 copies of each gene. 1 from the chromosome they inherited from their mother at fertilization, and 1 from the chromosome they inherited from their father at fertilization. So if the DNA sequences of these chromosomes, we blew those up a little bit, was ATG, although chromosomes are much longer than 3 nucleotides, and if that one was ATC, the reason that geneticists determine that these are homologs, homologous chromosomes, is that the DNA sequences of the 2 chromosomes are very similar. That's how we know they're both copies of chromosome 1. But there can be the occasional genetic difference because these chromosomes came from 2 different individuals, the mother and the father. And so when that happens, those genetic differences are combined together into the offspring. So offspring are genetically different from the parents, and that's the whole point of and purpose for essentially sexual reproduction. So there are 2 copies then of almost every gene in every individual's cell because there are 2 homologs, 2 copies of every chromosome. And there's a benefit to this, being diploid, that what happens if a mutation causes a particular gene to become non-functional, to lose its ability to produce a protein or to produce a functional protein or to gain a new ability that's maybe deleterious or detrimental to the cell. The great thing about being diploid is you've got a backup copy of every gene. So if you're in this situation, you might be in trouble if you're a haploid individual, if you never have a second copy of a chromosome, if you've just got one toilet paper roll, what happens when it mutates or runs out of paper? Diploids have that backup copy. So if something bad happens to one allele of a gene, one of the two copies, then you still have potentially the other copy, the other allele to rely on. Now, this brings up the concept of dominance, the fact that we can have two different versions of a gene, two different alleles in the same cell. And again, those are inherited at fertilization. So one copy of every chromosome that you and I have, and most living organisms, at least diploid organisms, came from their mother at fertilization. And the other set of chromosomes came from the father. So let's introduce this in a different style. So here is a way that some geneticists, many geneticists, diagram chromosomes as a single line. It's meant to represent a double helix chromosome. And then scientists and geneticists in particular will often diagram the positions of genes as right-angled arrows with an arrow pointing one direction. And that region, let's say, is the position of a gene on that chromosome. And the arrow typically indicates the transcription start site. That's the spot where RNA polymerase starts reading the DNA molecule and producing an RNA transcript, the first step in the central dogma. That defines part of this region as the coding region. That's the part of the gene that encodes proteins, amino acids that will make up the protein. And that leaves the other two regions that aren't coding region, that are part of the transcript, as untranslated regions. Those are parts of the gene that don't encode protein. Upstream, or to the 5' direction of the gene, is a regulatory region called the promoter. And this is where transcription factors and other sorts of proteins bind by recognizing different DNA sequences found in the promoter region of each gene. And that binding of proteins can either positively or negatively determine whether or not this gene is expressed, whether it's transcribed and translated and produce a protein. So each individual is diploid. They'll have two copies or homologs. Let's say the top one is the paternal and the bottom one is the maternal homolog. That's not standard genetic nomenclature, P&M. That's my own nomenclature. And let's say, what happens if the maternal version of the protein has a mutation, which tend to be indicated by asterisks or X's or some sort of notation. So let's say there's this mutation, and that is, in this case, inside the coding region. So what this mutation's effect could be, in this example, is that protein isn't functional. So maybe the gene is transcribed and the protein is translated, but the protein's amino acid sequence is different, and that means that the protein that this maternal allele encodes isn't functional. Okay, so this brings up this concept of dominance. So we have a protein produced from the paternal allele and a different, let's say, shape protein produced by the maternal allele. What might this do to the cell? Well, let's see. If the organism that has one wild type copy, sometimes abbreviated WT, that is the original or the normal functioning allele, and one mutant copy, there are a few different things that could happen. On one hand, the presence of some wild type protein or normal protein might be enough for the organism to continue surviving as normal. That is, this mutation might have no effect in this situation where there's one wild type allele and one mutant allele. On the other hand, this mutation itself, the presence of that mutant protein, could cause a negative impact on the organism. And so this all sets the stage for the analysis of the concept of dominance. That is, what happens in the heterozygote, that is the organism that has two different alleles. We want to know what the phenotype or the trait of that organism is compared to. Let's take the example of, say, the HOXA1 gene. There'll be two different alleles of that gene, but let's say one of them is genetically different, mutant, so we'll draw that in blue instead. So two homologs, two alleles of the HOXA1 gene. Black is wild type and blue is mutant. So there are three different combinations of these two alleles that could be present in any individual in that case. The individual could be, if they're diploid, which mice and humans are, they could be homozygous, HOXA1 over HOXA1. This indicates homolog 1, its allele, and then the allele of this gene on the other homologous chromosome. Chromosome, doesn't matter, we didn't specify which chromosome this was in this case. So this is a genotype notation. We can also abbreviate this more simply if we want to as black H/black H. That is the typical genetic nomenclature for writing a genotype for a diploid. One allele / indicates that we're switching to the next homolog, and then we write the genotype or the allele on the next homolog. Then we could also have individuals that are blue H over blue H, because all possible allele combinations exist. That's one of Mendel's laws. And then the third possibility is that we could have an individual like the one that's drawn here that is blue over black or black over blue. The order doesn't matter. So here's the next important part of genetic nomenclature is the terminology for these three different genotypes. The two that have the same allele on both homologs, regardless of black or red, are called homozygous. Homo meaning same. And the one in the middle, which has two different alleles, is the heterozygous genotype. So there's one heterozygous genotype, and there are two different homozygous genotypes. And the way we know whether or not dominance exists in the relationship between two alleles is what the trait is of the heterozygous individual. Is the heterozygote like this parent, for example, or this genotype? It doesn't have to be the parent. Or is the heterozygote like the blue homozygous parent? So there are a few different possibilities here. There's a possibility that there's simple dominance. And so, for example, if the trait that we're looking at is, well, let's see. What's the trait that we should talk about here? I guess we could do pigmentation. That's not what Hox genes would produce, but since I'm using colors, let's indicate that if there's simple dominance, the individual that's blue H over blue H is colored blue. And the individual that is black black H over black H, I can't draw this on top of that notation, is a black pigmented individual. So if there's simple dominance, if the blue allele, let's say, is recessive, we would be defining that as the phenotype of the heterozygote. So if blue is recessive, then what's the trait of the heterozygous individual? Black is dominant if blue is recessive, so this individual would be black if blue is recessive. Now, there are other possibilities as well other than simple dominance. So we've got codominance, and we've got incomplete dominance. So we've already just looked at simple dominance. We've looked at the phenotype of the 3 different genotypes, and we see that the heterozygote has the black H-like trait. That's what defines black H as dominant, and by extension, by definition, then blue H is recessive to black H. In codominance, what happens? In codominance, we get a mixture of the two. That is, we might get an individual that's half black and half blue. That is, both traits appear distinctly in the organism. The other possibility here, other than dominance, simple dominance and recessiveness, and codominance, is incomplete dominance. And that would be a situation where, although I'm not very good at colors because I'm colorblind, I'm not going to try. If the heterozygote is a blend of the 2 parents' traits. So for example, what's a blend of black and blue? Some sort of murky gray? I don't know. So the idea here is that there would be a blend of colors. This is more often in textbooks indicated in crosses between flowers, where you might have a red parent and a white flowered parent, and their offspring is pink. It's a blend of the 2 parental traits. That is an example of incomplete dominance. And so all dominance tells us, and the way we find out about dominance is, again, to reiterate, by comparing the 3 different genotypes and looking what the mixed allele combination, the heterozygote, looks like. Does it look like the black over black or the blue over blue? Or is it a mixture of both, or is it a blend of both? And that can help us distinguish the different dominance traits. Now, unfortunately, things aren't always this simple. So turning our attention back to this situation in mice. Mice are diploid. They have 2 copies, 2 alleles of every gene on their 2 homologous chromosomes. But now let's think about what happens when you have more than 2 copies, or more than 1 copy of a gene, more than 2 alleles of a gene inside the same cell. And this is the situation of paralogs, where genes that have duplicated have multiple spots in the genome where essentially the same gene is found. Now we have more than 2 copies, almost, essentially, of the same gene. So for this, I'm going to describe the situation of the HOX genes, and I'm going to pick in particular on the HOX4 gene. So in mice, there are 4 different paralogs of the HOX4 genes. There's one called the A, which I don't know exactly where on the chromosome it is, but I'm going to say it's near the telomere. On chromosome 6, we find HOXA4. So this is the A cluster. This is where all the HOXA genes are, HOXA1, 2, 3, 4, etc. On chromosome 11 are the HOXBs. And on chromosome 15 are the HOXC4 alleles. And then on chromosome 2 are the HOXD4 alleles. So each different group, each different homolog has a different paralog. So these are 4 different paralogs of each other. HOXA4 is a paralog of HOXD4, B4, and C4. So there are 4 different paralogous genes, and each of those 4 has 2 different alleles potentially, because there are 2 alleles of HOXA4, 2 alleles of HOXD4, 2 alleles of HOXB4, and 2 alleles of HOXC4. So that's a total of 8 different alleles of the gene, the ancestral gene, HOX4. HOXA4A from mom, HOXA4B from dad, HOXD4 from mom, HOXD4 from dad, and so forth. So that means there are a lot of copies of this gene inside each cell. And the point of the Wellick and Carpecci paper is to ask, for Hox genes, which are involved in development, how many of each paralogous group, all 8 paralogs, their alleles and their paralogs, how many of those does a mouse need to develop normally? In other words, what was the fate of these duplicate genes? There are 4 duplicate sets of genes, 4 paralogous groups. What happened to them? Are they redundant? Or maybe was there some neofunctionalization? Or maybe was there some subfunctionalization? So for each different Hox gene, A, B, C, D, those different paralog groups, for HOX1, HOX2, HOX3, HOX4, HOX5, we could go through each gene and ask, what was the fate of the duplication? And that is the point of the Wellick and Carpecci paper. So this is the title of that paper, and here's an example of one figure. So I want you to understand what this figure represents before you read the paper, because almost all of the figures look like this. So it's really important to understand what exactly this is that we're looking at. This is a view of the vertebrae of a mouse. So the tissue has been removed and the bones have been stained, maybe with alizarin red, for example, so that we can see the ribs, the vertebrae. Here are the lumbar vertebrae and which are the sacral vertebrae. And this individual also has these different panels, panel G, panel H, panel I, panel J. So importantly, we need to figure out what those pictures are. What are those pictures of? Well, T13 is a vertebra, and T12 is the one next to T13. So they've labeled which vertebrae each of these panels represent. The question then is, how did they take this picture? What did they do? Well, imagine if a camera was placed at the tip of the tail and was photographing up the vertebral column. That's basically what these images represent. They're cross sections. They're taking this vertebra and looking at it edge on or end on. So we can see a cross section of the vertebra and the extension then of those ribs. So we can do the same thing for different vertebrae. And then what the authors are going to do is to compare the phenotypes of of mice, my bad habit, with different genotypes. Because again, they want to know what's the redundancy of these different alleles and paralogs, not alleles, what's the redundancy of the paralogs of the different Hox genes. So this is a control. It's wild type. It's got the typical genotype of whatever mouse line the authors were working with. So in the paper, they have to compare the phenotype or trait of a wild type mouse to a mutant. So what is this mutant we're looking at in figure 1? These are just 2 panels from figure 1. So this indeed refers to HOX10, and then the letters indicate the genotype of this mouse. And in this case, they've left off the slashes that I mentioned earlier. Normally, we would write this as HOX10, little a over little a, and little c over little c, and little d over little d. Because we know that the 4 different HOX clusters, A, B, C, and D, reside on different chromosomes. So it's appropriate to say the genotype of HOX10A is little a over little a. And the genotype of HOX10C, which is on a different chromosome somewhere else, is C over C. But they've just simplified the notation in this panel. Last thing for now is that the lowercase letters, in this case, don't really mean anything. They're not trying to indicate dominance versus recessive, which sometimes is conducted using capital and lowercase letters, but not always. In this case, this simply indicates that this is a mouse that's lacking both alleles of A, of C, and of D. In other words, these scientists have removed both copies of the HOX10A gene, both alleles, and they've also mutated both alleles of HOX10C, and they've mutated both alleles of HOX10D. And now here, they're just asking, what does that do to the development of the mouse? And then we use our eyes and we compare the top and the bottom panels, and we see what the effect is on a mouse of not having these 3 paralogs of the HOX10 gene, HOX10A, HOX10C, and HOX10D.